What if I told you there's a new groundbreaking genetic engineering tool that could change our world and the living things within it in countless ways? This tool is called CRISPR-Cas9, or CRISPR for short. CRISPR itself wasn't created by scientists. It was found in the natural world. See, the CRISPR-Cas9 process is the immune system of bacteria. But what does CRISPR mean? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. The short palindromic repeats refer to repeated strands of DNA, about 20 to 40 letters long, arranged in a palindrome. A palindrome is when a pattern reads the same left to right as it does right to left, like taco cat. The clustered regularly interspaced pieces are strands of DNA that go in between the palindromic repeats. Okay, now imagine I'm inside a bacterium. I'm going to use these Krispy Kreme donuts to demonstrate the CRISPR process. So these glazed donuts here represent the short palindromic repeats. When a virus invades the bacterium, a CRISPR-associated protein, or Cas for short, swims over and cuts out a segment of the virus's DNA. This Cas protein then inserts the segment in between the palindromic repeats, creating a clustered regularly interspaced piece. The viral DNA is copied into RNA, which is then attached to a Cas9 protein. Now, this specific Cas protein is in charge of searching for invading viruses that contain matching DNA to the RNA inside it. Basically, this Cas9 Pi server will look for any viruses that contain chocolate sprinkle DNA. Look out! Another virus is invading the bacterium. Luckily, the bacterium is ready this time, and the Cas9 protein inspects the viral DNA. It's a match. Cas9 attacks, destroying the viral DNA by cutting it with a chemical reaction. Each time a new virus enters the bacterium, its immune system goes through this process, placing segments of viral DNA in between these palindromic repeats. CRISPR becomes a sort of library that contains information about all of its enemies. I can hear you asking, how does this relate to genetic engineering? Well, scientists figured out how to hack this CRISPR system in order to target any DNA, not just viral DNA. These glazed donuts now represent the DNA inside a cell. First, the scientists identify the gene that they want to edit, in this case, the donut with sprinkles. Then they design a guide RNA, or gRNA, that matches the gene and attach it to a Cas9 protein. Now, instead of searching for viral DNA that matches the RNA, Cas9 will search for the specific DNA of the scientist's choosing. Once Cas9 finds the DNA match, it slices through the DNA. Now, the cell will try to repair the broken DNA, but the cell can make mistakes, and the gene is often unusable and turned off. Scientists can use this to their advantage by targeting a gene that they want deactivated. They can also cause the cell to perform a different repair process if they add a sequence of template DNA to CRISPR, which will repair a defective gene or add a new one. CRISPR has the potential to change our world for the better. Thanks for listening.